Hi, this is Brandon. This is my submission for the DNA model project. So to start off, we have the structure of DNA. Now, the shape of a DNA strand like this is a double helix. So it's a very distinct shape, like how it curves, and will still bounce back up. It's a very distinct and unique shape. Now, the DNA strand itself is composed of several nucleotides, which make up both DNA and RNA. Deoxyribonucleic acid is what DNA stands for. It's essentially what DNA is also comprised of, along with the nucleotides. Now, DNA is made up of 4 nitrogenous bases. Now, they are identified in 2 groups, pyridamines and purines. Now, the pyridamines are cytosine and thymine, while the purines are adenine and guanine. Now, you can see they're identified by T right here. The green is T, or thymine, A for adenine, C for cytosine, and orange or G for guanine. Now, they are paired based off a process or idea called hydrogen bonding, which essentially it depends on how they pair. So C will pair to G, or A to T, and so on. And it's always that. So T will to A to T, C to G, G to C. Now, there's also phosphoric acid in DNA. Now, the sugar in DNA, which is the sugar in the DNA molecule, is deoxyribose, and the one in RNA is ribose sugar. This is the phosphates based on the inorganic compound phosphoric acid. Now, genetic code is the way that the 4 bases of DNA, A, C, G, and T's, are strung together in a way that cellular machinery, the ribosome, can read them and turn them into a protein. And essentially, that's how they code. That's what makes DNA unique, is how they code in these pairs of 3, which are a codon. That's what makes DNA unique, and each codon would code for a trait, making each trait unique in a person. Now, the chromosome structure of DNA is DNA tightly coiled many times around proteins called hysteres that support its structure, which is why it is a double helix shape as well. In copying of DNA, when DNA copies, it is semi-conservative, meaning you will have 2 old strands, or original strands, break off, and then from that, it will use hydrogen bonding to go ahead and form 2 new strands, make it semi-conservative. Because the 2 new strands each have a little bit of old, a little bit of new, so semi-conservative of the original. Now, a leading strand is a single DNA strand that during DNA replication is replicated in a signal direction by the replication fork. DNA is added to the leading strand continuously, one complementary base step. So when you have a replication fork like this split off, each of these new strands has a leading strand, which essentially will go continuously pairing by the bases. So T to A to T, C to G, and so on. RNA primer is a short nucleic acid sequence that provides a starting point for DNA synthesis. In living organisms, primers are short strands of RNA. The primer is removed before DNA replication is complete, and gaps in sequence are filled with DNA by DNA polymerases. A lagging strand is the strand of new DNA whose direction of synthesis is opposite to the direction of growing replication fork. Because of its orientation, replication of lagging strand is more complicated as compared to that of the leading strand. Now, when splitting, DNA forms a replication fork, as you can see right here. It almost forks out, creating this unique shape at the end of it, and it forks out, making it have that 2-pronged shape. Another process that involves DNA is transcription. But in transcription, we see DNA turn into RNA. But in order for that to use it, we need something called mRNA, or messenger RNA. But in that process, mRNA does not have T. Instead, T becomes U, or uracil, which is another one of the bases, a 5th one, per se. RNA polymerase synthesizes RNA by following a strand of DNA. RNA polymerase is an enzyme that is responsible for copying a DNA sequence into an RNA sequence during transcription. Now, in translation, you have transfer RNA, which is a type of RNA molecule that helps to decode the mRNA, or messenger RNA, into a certain type of protein. And then these proteins essentially are made up of things called amino acids, which is an organic substance made from certain combinations of bases or codons in order to code for DNA. So an amino acid would be maybe the combination of AAC. That would code for a single type of amino acid. or what the amino acids are also proteins, which all have their own unique names based off of the trio or codon that forms the protein. The ribosome is a small particle consisting of RNA and associated proteins found in large numbers in the cytoplasm of cells. They bind messenger RNA and transfer RNA to synthesize proteins. And that's all.